Now that we know the structure of DNA, let's talk a little bit about what it can do. Genome functions really boil down to one thing: the central dogma of molecular biology. It is that important that we have this really kind of hoity-toity name for it, the central dogma. We start with DNA, and then through a process called transcription, we copy a portion of that into mRNA or messenger RNA, and then in a process of translation, we turn that mRNA into a protein. And this is how all proteins in our body are made. Another way to talk about this is gene expression. This is making proteins from genes. So really, this is all different words to say pretty much the same thing. But let's take a little bit more of a close look at what we're going on here. Here we have a eukaryotic cell, and within the nucleus, that is where our DNA is housed. DNA never leaves the nucleus because we want to make sure it's protected so it doesn't get corrupted or broken. Because remember, it's important to keep it intact so we can reliably transmit hereditary information from a parent to its offspring, whether we're talking about that on the organismal level or just, cells within your own body. Because remember, when that goes wrong, that's cancer. So, in our nucleus, that's where transcription happens. We make a copy of a small portion of our DNA and turn that into mRNA. That mRNA then leaves the nucleus, and that's where some more exciting stuff happens. It goes to a ribosome in the endoplasmic reticulum, and that is where it goes through a different process called translation, where we take that code and turn it into proteins. The way we do that is by using the genetic code, which is visualized here. We use a sequence of 3 base pairs to use this code to translate that short sequence into a different amino acid. So here you look at the first letter. So in our example, we have our first letter A, our second letter is C, and then our third letter is G. It can be a little bit complicated to use this until you're used to the format of our genetic code, but that's because it's hard to demonstrate 3 separate axes on a 2-dimensional piece of paper. What we call this is a codon. A codon is that sequence of 3 nucleotides of DNA or RNA, depending about whether you're talking on the original DNA template or that mRNA that we actually do the translation with. And this is our unit in the genetic code. You can see here in the ribosome, our DNA actually kind of feeds through it, and we have a transfer RNA that brings the appropriate amino acid because that transfer RNA will match with a 3 base pair codon, and then we just, add on another amino acid, kind of like beads on a string. The next important thing our DNA can do is replicate. This is important because if DNA couldn't replicate itself, well, we'd never have another cell because we need this DNA as our template in all of our cells so we can make proteins in every single cell. So what you can see here is we actually unwind the DNA, and then from each of those strands that are separated, we just copy new strands. And this goes back to that complementary base pairing, and that's why it's so important. Without that complementary base pairing, we wouldn't be able to reliably make new copies. But since we do have that, as long as you have a single strand, you can replicate it pretty much exactly. So if you've ever heard that cheesy pickup line, "I wish I was DNA heat click case so I could unzip your genes," that's what it's talking about. And don't use this line on me. So we see this happen both in mitosis and meiosis because we are replicating the genome in both cases, though they do happen in slightly different ways. In meiosis, we actually have 2 rounds first. We replicate the genome, we separate the different chromosomes, and then from there we separate them even further. So all of the 8 daughter cells that we create only have half of the full complement of a gene. But one of the cool things that can happen in meiosis is called crossing over. This is the exchange of genetic material between homologous chromosomes during the first prophase of meiosis, and this is one of the mechanisms for genetic recombination. Remember, we have 2 copies of each gene, so everyone has 2 copies of every chromosome in your cells. So here, when a chromosome duplicates, we have these sister chromatids, and then we have a homologous pair. So we've colored them with different colors here so it's easier to see, but imagine like these are both copies of chromosome 1 per se. Then these homologous chromosomes will cross over, They will exchange some of that genetic material, and now our resulting chromosomes are a little bit of a patchwork. So this is how you can get, some of a chromosome from your grandpa, but some of that might actually be from your grandma because of this crossing over here. And what's cool about meiosis is there's a couple different ways that we can increase genetic variation. Of course, there's the crossing over, which we just discussed. Then we have random assortment. We don't know which of those chromosomes that you will get from your dad or your mom. And also mutation. If mutation happens during meiosis, that mutation will be passed on to their offspring. So, what functions does DNA do?